Thank you so much for inviting me to this. I've been hearing about this this seminar series. It seemed like a wonderful kind of pandemic innovation, and I'm glad to see that it's still going on. And I'm really excited to talk to this group. Okay, so let me dive in. So today I'm going to ask the question, how do we learn across timescales? So we need to learn both slowly across many hundreds or thousands of learning opportunities, and that might unfold over, days and weeks and months and years. But also often we need to learn quite quickly, sometimes within a few exposures or even after just one exposure to some piece of information. And that faster timescale of learning is especially important, of course, when your environment changes in some way, and you need to learn quickly how to behave adaptively in that environment. So if you are used to the streets of Philadelphia and you get off the plane in Tokyo, you're going to have to learn very quickly that you should be looking to your right when you're crossing the street. So trying to quickly learn new information directly in the neural populations that represent related existing knowledge has the danger of overwriting that existing knowledge. So to the extent that you're going to try to use the same neurons to represent the new stuff and the old stuff, there's this kind of infamous problem that you have to face that you might cause retroactive interference. And this is of course, a very deep, difficult problem for neural networks that use distributed representations. And one elegant solution to this problem was proposed by the complementary learning systems theory, which said, well, okay, so you shouldn't try to learn the new and the old stuff in the same neural populations. You should have this buffer where you encode new information in the hippocampus using these kind of pattern-separated representations that avoid interference amongst those new experiences. And then the hippocampus can replay those experiences offline, maybe especially during sleep, and that would allow the neocortex to slowly accumulate the statistics across those experiences in the way that allows you to ultimately build up that nice distributed representation that will allow you to see how everything's related and generalize. So the idea is to kind of take the non-stationary distribution and turn it into a stationary one through this interleaved replay. So I think that the interleaving aspect of it is, I think, a sometimes underappreciated aspect of the original theory, but that was really the key to how you can build up this kind of robust, stable representation in cortex. Okay, so right, you avoid interference by not writing information directly to that final store. So that framework, I think, has been very useful. I think it explains a lot about how our memory systems interact. But to solve the complete puzzle of how we learn across timescales, I think there's a few more questions that we will need to answer. So one obvious question is, how do we learn across episodes quickly? So the complementary learning systems told us how you learn like an individual kind of snapshot quickly. But maybe it takes you several times crossing the street before you get used to the flow of the Tokyo traffic. And how do you aggregate across experiences quickly on that timescale? Another question is, where and when can learning occur directly in neocortex? So what kinds of information has to go through that process of initial hippocampal storage and then offline replay? And what kinds of information can you incorporate directly into neocortex? And then how might sleep and replay actually serve to integrate hippocampal and neocortical learning? This story wasn't really fleshed out in the original framework, and this is one of the focuses of my lab. Okay, so just to give you the kind of the take-homes in advance, I'm going to make two claims. One is that we can kind of make progress towards understanding those questions by noticing that the hippocampus is not homogeneous itself. And we propose that there are rapidly learned distributed representations in the hippocampus that complement the classic kind of pattern-separated representations and help us explain how you can learn across experiences quickly. And then the neocortex is not homogeneous either, of course. And we're going to propose a kind of a broader gradient here where it's not, it's not just hippocampus versus cortex, but actually a kind of gradient of areas running from early sensory areas up into the hippocampus of increasing sparsity and increasing plasticity as you go. Okay, so let's start with the first question. How do we learn across experiences quickly? So here's my one slide on my PhD work. So we had been running these experiments where participants were seeing streams of abstract images presented one at a time with some hidden temporal structure. So in this study, there were pairs of images that always occurred together in the sequence. And we found that over the course of like an hour-ish of exposure to these sequences, the hippocampus came to represent the statistically associated objects more similarly. So from before to after learning, you see this increase in pattern similarity in the hippocampus. So that is, so that suggests, the hippocampus is sensitive to these kinds of statistics on this time scale. And it suggested that it was using this kind of overlapping code that is not what you might have expected from the classic pattern separation story. And then it wasn't just simple pairwise statistics. It also was true for more complex, higher order kinds of structure, like this temporal community structure. So here, the shapes from the same community came to be represented more similarly in the hippocampus. And then it wasn't just that the hippocampus is sensitive to these statistics, but also that we think it's kind of causally necessary for, expressing behavioral sensitivity to these kinds of statistics. So patients with hippocampal damage don't show any behavioral evidence of this kind of rapid statistical learning. So this got us thinking that the hippocampus is going to be a key region for thinking about this across episode rapid learning. But then we had to figure out how to reconcile those observations with the long and very robust literature on the role of the hippocampus in pattern separation and keeping different episodes separate to avoid interference. So how do we think about those two different kinds of representations as living in the same area? And the way that we've been thinking about this is through use of this neural network model of the hippocampus that we call Seahorse that implements some of the properties and connectivity of the different subfields of the hippocampus that we think might be important for explaining its function in these domains. So this model has entorhinal cortex that is going to process the input and output. And then it has three hidden layers that represent three of the hippocampal subfields. So dentate gyrus, CA3, and CA1. So there are two main pathways through the hippocampus and in this model. There's this trisynaptic pathway that runs from entorhinal cortex to dentate to CA3 to CA1. And this is the pathway that is famous for this pattern separation function. So there's this very sparse connectivity into dentate gyrus that we think is crucial for that orthogonalization kind of function. So you're taking very similar inputs in entorhinal cortex and projecting them to relatively distinct populations of neurons in dentate gyrus, and that's going to allow you to keep track of each of your individual episodic experiences. So that's the trisynaptic pathway. But then there's this pathway that runs straight from entorhinal cortex to region CA1. That's the monosynaptic pathway. And this pathway seems to have some different properties. So it has less extreme pattern separation. That's probably related to differences in its connectivity. It also seems to have a slower learning rate than the trisynaptic pathway. So we're very inspired by studies like this in rodents that show that if you lesion specifically the CA3 to CA1 pathway, you actually still can do incremental learning, like Morse water maze learning. You knock out the ability to do like one-shot fear conditioning kind of learning. So you're going to need the trisynaptic pathway for that one-shot learning, but the monosynaptic pathway actually can on its own support learning. It's just a little bit more incremental. Okay, So we have shown in old simulations that I'm not going to go through today, you can take my word for it, that these two different sets of properties are very well suited to these two different functions of episodic memory. I mean, there's a long lineage of models showing that this pattern separation function in the trisynaptic pathway is very well suited to one-shot episodic memory. And then our kind of new piece of this is that the slower learning and the more overlapping representations in the monosynaptic pathway we think are very well suited to this kind of rapid statistical learning that we had been studying. So the claim is that there's a kind of a microcosm of the broader complementary learning systems dynamic playing out within the hippocampus, where the trisynaptic pathway is handling that fast learning on pattern separated representations, and then now the CA1 is like a little piece of neocortex sitting in the hippocampus that's doing the kind of slower learning with building up these distributed representations that are going to allow you to see how things are related across experiences. Okay, so we understood how those properties would allow us to explain those temporal statistical learning experiments that we were running. But if the model is right, then this monosynaptic pathway learning strategy should allow the hippocampus to support many forms of across episode structure learning. So any situation where you're trying to pick up on structure across experiences, this kind of strategy might be useful for. And so we've been exploring kind of other domains where we think that this might be relevant. And one domain that we're very interested in is new category or concept learning, because this is also a situation where you need to pick up on structure across experiences. In this case, the structure across exemplars from a category. And we know that the hippocampus is important for this kind of learning. So first we thought, okay, so let's see how this model performs different kinds of canonical category learning tasks. So I'm going to show you a couple examples of those simulations. Excuse me, sorry. So the first is the classic weather prediction task. So in this task, you see a set of abstract cards and you have to predict sunshine or rain. And we know that amnesics are able to do this task to some extent. They're not at chance, but they are impaired relative to controls. So we don't think that the hippocampus is the only region involved in this task, but we think it's contributing something. So the question is, what might that be? And when we run this task in the model, what we find is that, so here's the categorization performance. The green is the regular intact version of the model. The orange is a version that only has access to its monosynaptic pathway. And then the purple is a version that only has access to its trisynaptic pathway. And you can see that the model performs similarly to people. And it's really the monosynaptic pathway that's driving its ability to do the categorization. But if you change the assessment of the model, so if you say, instead of categorization, can you remember particular configurations of cards that you were shown during the training, then the pattern flips, and now the trisynaptic pathway is better at that than the monosynaptic pathway. So both of the pathways are learning from the input, but they're learning something different about the input. I'll show you one more example. This is a paradigm that we have used empirically in the lab. This will come up again later in the talk, where we teach people about these three categories of novel satellite objects, where the satellites in the same category share most of their features, but they also have unique individuating features and a unique name. And we had found in one of our fMRI studies using these stimuli that CA1 represents this category structure, but not dentate and CA3, meaning that the satellites from the same category are represented more similarly than satellites from different categories. And what we find in the model is that so this is generalization performance, meaning you see a new satellite that you haven't seen before, but belongs to one of the categories that you had learned. Can you figure out what category it's from? And we find that the model's good at this. And actually, it's really even better, much better, if you only allow it to have access to the monosynaptic pathway, and then the trisynaptic pathway struggles with generalization. But again, if you change the assessment, so if you say, to what extent can you remember those unique individuating features of these very similar looking objects? In that case, the trisynaptic pathway is able to do that, and the monosynaptic pathway really struggles with that. So the trisynaptic pathway is useful here to the extent that you are trying to keep track of those arbitrary details. But it's really the monosynaptic pathway that's understanding the category structure. And the internal representations in the model are what you'd expect. So CA1 shows that nice category structure, whereas dentate and CA3 keep everything orthogonal. And you can see that, kind of matches what we had seen in our fMRI experiment. So I mentioned that there's this kind of trade-off here where it's actually better to silence the trisynaptic pathway if you're going to attempt to do generalization. That suggests that it would be useful if there were a way to kind of control which of these two pathways is driving your behavior, depending on the task that you are currently working on. So it's this kind of like average of the two pathways actually serves surprisingly well in a lot of contexts, but there are situations like this one where there's really a trade-off between the details and the kind of the abstraction. And so we've gotten very interested in kind of, might there be a way of controlling which of these two pathways is influencing behavior? You can see the trade-off is also happening here in the weather prediction task as well, although not as strong as in this paradigm. So this is a new line of work, but I thought I would just mention that we are now exploring this control question in a mixture of experts setup. So we've ported our model into PyTorch, which is great because I think there were some people who were willing to download Emergent and try to use it, but I think it'll be much more easy for people to download and play with now that it's in PyTorch. So we have a kind of abstracted version of the model that you can play with in PyTorch. And one of the nice properties of it is that it's now, it can handle real input. So the previous versions of the model were kind of constrained to our little hand-coded inputs of the stimulus structure. But now you can take whatever your favorite ConvNet is and stick it in front of this model, and it can handle that input nicely. And so we can give it real images, which is great. And here we have this module that we're calling the MPFC that we're going to allow to kind of learn how to deploy these two pathways as a function of the current task. So here we're training the model on fashion MNIST, and there's two different tasks. One is the regular categorization task. The other is an exemplar memory task. So can you remember the individual specific sweater or whatever? And the model is going to try to learn to deploy the right task the right pathway for the right task. And we know from the rodent literature that MPFC has the right connectivity and has the right influence on the hippocampus to kind of allow for the potential for this kind of task-dependent modulation of what's happening. And so we thought that was a good candidate region for this control. And we find that, without the mixture of experts learning, we get that double dissociation between the monosynaptic pathway and the trisynaptic pathway on category learning versus exemplar memory. But if you allow there to be this control system that can decide what to deploy at the right moment, it can do very well in both tasks. So we're excited about exploring this more and seeing if it can explain some of the human findings on MPFC hippocampal interactions and some of the rodent findings. Okay. All right, so I said that category learning was one kind of task where we thought that the monosynaptic pathway strategy might be useful. I'm going to give you one more example of a kind of task where we think that this pathway might be relevant. And this is associative inference. So here you learn that A and B go together, you learn that B and C go together, and then you have to, at test, make this transitive link from A to C. And we know that the hippocampus is important for this ability from rodent lesion work, and it comes up in lots of human fMRI studies. So in the literature, there's been several different proposals for how the hippocampus might be solving associative inference. I'm going to kind of contrast two of those proposals here. So one proposal was implemented in this Vermerge model. And this model said, we are going to kind of double down on the pattern separated conjunctive kind of representations in hippocampus. And we're not going to allow for there to be any overlap in these different memories as you're learning about them. But you can still solve this problem through recurrent computation at retrieval. So the idea is that if you've stored your separate AB and BC memories, you can still get from A to C at test if you have kind of set up your connections to allow you to do the recurrent computation. So you can get from A to AB to B to BC to C at test. That's very different than the strategy that I've been talking about, which is, you could do interleaved learning to build up a distributed representation that directly represents the overlap across these related experiences. So here the idea would be you have your A and your C representation. Maybe they start off with some overlap. And then as you go back and forth between studying A, B, and B, C, there's pressure to push those representations closer together because they share that B item in common. And then that makes the inference at retrieval time totally obvious and automatic because A and C are just sharing neurons. So the idea is that this type of strategy should be really fast and automatic, but dependent on having experienced the pairs in interleaved presentation order. Whereas this kind of recurrence at retrieval strategy, it's slower and more effortful at test. You have to go through all your recurrent computations, but it's not dependent on the presentation order. So this kind of representation is not susceptible to the retroactive interference problem we talked about, whereas the distributed representation is. And so we thought we would take these different properties of these different strategies into a behavioral paradigm where we might be able to try to separate which of these things people are using. So the idea here is to present participants with the kind of typical associative inference triads, but some of the triads will be presented in blocked order, meaning that you'll see all of the ABs before you see any of the BCs. Sorry, that's the blue one. So like this one, all of the ABs before you see any of the BCs from that triad. And then there will be other triads where you alternate between the ABs and the BCs over the course of training. And then at the end of that exposure, we have two different tests. One is a speeded recognition test where we ask people to quickly make a judgment about whether they studied these two objects together directly. And the answer in this case is no. So even though these two objects have this indirect relationship through the orange object, they were never studied directly together. So here you'd say no. And then at the very end, we do the classic, like the regular explicit inference test, where we show people an object and we say which of these two objects below is associated with the object above. And we give them plenty of time to make that decision however they would like to. So we don't predict differences between the conditions in the explicit inference test because we think you can use either of those two strategies to solve that problem. What we're really interested in is the speeded recognition test. And that's because if you're shown a trial like this, where you have these two indirectly related objects, and you've built up this directly overlapping representation between A and C, then this judgment becomes confusing because it's going to be harder to say, no, I didn't study A and C together if you've built this directly overlapping representation of them. And so the prediction is, if this order manipulation matters, then you should be slower to say that the interleaved ACs were not studied together relative to the blocked ACs. And maybe you'll even false alarm and say that you've studied those things together when you had not. Okay, so here's the results for the explicit inference. So we didn't predict a difference between interleaved and blocked, and indeed we do not see a difference. And so these are like 3 variants of basically the same task, small changes. So you can think of them basically as replications. The third one is especially well powered. So you can see there's really no difference between interleaved and block conditions there. But in the speeded recognition case, we see that people are slower to judge that the interleaved ACs were not studied together, which we think is consistent with this idea that in the interleaved case, you're building up this more overlapping representation of A and C that makes those particular judgments more confusing. And in 2 of the 3 experiments, we found that people actually false alarm. So they'll say that they studied those things together when they never actually did. So that's an example of how the kind of the overlapping representation is not useful, right? It's like confusing you about what you studied. But of course, we think that that kind of representation should be useful for certain kinds of situations like generalization. So we ran a couple experiments to try to show off like what that kind of representation might be good for. So here we did the same exposure as before, but then at the end we taught people that some of the A objects have a particular property that they have to remember. And then we asked how likely are they to generalize that property to the indirectly associated C object. And we found that they were more likely to do that in the interleaved case than the blocked case. And then we thought it would be interesting to turn the experiment into a statistical learning paradigm. So instead of presenting the pairs in these kind of like discrete demarcated episodes, now we're going to show the objects one at a time, and they have to figure out from statistics over time, which of these objects are paired together. And this is the kind of situation that is really quite difficult for the remerge strategy to handle because it struggles to kind of parse noisy input over time, whereas the distributed representation is very good at that kind of thing. And in this case, we find that even in the explicit inference, where we give people plenty of time to make the judgment, now there's an advantage in the interleaved case. And actually, if you break it down separately for interleaved and blocked, the blocked condition is not even above chance. So in this kind of situation where you're picking up on statistics over time, you really need to have interleaved exposure in order to pick up on the structure. Okay, so the take home so far is that we think we're accumulating some evidence that the hippocampus contains these distributed representations in the monosynaptic pathway that are a complement to the classic pattern-separated representations in the trisynaptic pathway, and that might allow for this kind of rapid statistical learning and generalization on this timescale of minutes. Okay, so so the work so far is focused on, rapidly learning novel structure where your prior knowledge has no bearing on learning the particulars of the structure that you are exposed to right now. But of course, a lot of our experience is not like that. A lot of our experience is kind of making small tweaks to our existing understanding of the world. And we wanted to ask how these processes that we've been studying relate to learning relationships in an environment that is representative of the distribution of your past experience. And so we're still interested in, the role of the hippocampus and region CA1 and that kind of learning. But when you're doing this kind of small updates to prior knowledge. We're also interested in the possibility that those regions that represent the prior knowledge might be able to kind of directly incorporate this new information. So this is a study to try to understand whether there are situations where you can kind of directly shape those cortical representations. So for this study, we used the natural scenes dataset, which is a really amazing dataset where 8 participants each completed at least 30 sessions of high resolution 7T fMRI. They viewed 11,000 images of natural scenes. So they look like this. They're labeled with the objects that are present in the scenes, like the giraffe and the bus and the coffee. And these objects occur in the scenes in the way that you would expect them to, because these are just real natural images. So we think of them as like representative of the distribution of your past experience. So first, we built an object encoding model that allows us to predict the voxel activity patterns for each of the individual 80 objects that appear in these scenes. And we did this by regressing each voxel's activity for each image against a vector indicating which objects were present in each image. And we found that this encoding model is successful. This is not new. Lots of people have done this kind of thing, but I will note that we were quite happy that this worked within the hippocampus. So it works, across all of visually sensitive cortex, but it also works in the hippocampus, which I don't take for granted because often our signal is not that great and object representations are not that clear. But with this very high quality data set, we do get these nice object representations in the hippocampus and much of the brain. Okay, and then within those regions that have those representations of the objects, we wanted to kind of restrict to the areas where we think there's some semantic content to those object representations. And to do this, we related the representational similarity that we saw in those kind of visual object representations to the similarity structure from word to vec, which is a measure of semantic relatedness from word associations and text. And the idea is that if you see this relationship, there's probably, it's bringing you out of the visual domain, probably into something more semantic. And we find that there's a kind of a subset of regions here that represent those more semantic objects, aspects of the objects. So a lot of ventral visual stream more anteriorly, and region CA1 represents this structure, which is interesting to us, and then the parahippocampal cortex very strongly. Okay, so we have these 30 sessions of data. So we can ask, are these object representations stable over those 30 sessions, or are they changing over time? And so here, these are plots of the of objects' similarity to themselves over time. Each of the dotted lines or dashed lines is an individual subject, and then the solid line is the average. And you can see that the lines tend to be a little bit negative. So that means that over time, the object representations are changing. And we find that there's this drift across sessions in region CA1, in parahippocampal cortex, and in perirhinal cortex, but it's especially strong in parahippocampal cortex. We tried V1 as a control just to kind of reassure ourselves that this early perceptual area has stable object representations, and actually there's debate in the literature about whether V1 representations really are so stable, but in this kind of analysis, they appear stable, which is kind of useful as a control for us. Okay, so if you do the whole brain version of this, you can see it's this more anterior subset of those semantic areas that are showing that drift over time. So some of those object representations are stable, and then some of them are changing slowly over time. Okay, so we don't we don't know what the cause of that change is. It could be just endogenous drift. It could be related to learning and your experience in the environment. It's hard for us to know. But we can ask whether there's any change that happens within an individual session, so on an even faster time scale. And in this case, we might be able to say something more concrete about what drives the changes. So if you see any changes in those representations on that very fast time scale, does it have something to do with what people are seeing in that session? And the way that we looked at this is to ask whether the ways that objects happen to co-occur in the first half of one of these sessions, does it predict the representational similarity structure in the second half of one of these sessions? So we take the co-occurrence statistics in the first half, we correlate them with the representational similarity of those object representations in the second half of a session. And we have a very strong control, which is to do the opposite. So are the representations in the first half of the session related to the co-occurrence statistics in the second half? And if that's true, then we're not looking at a kind of a causal effect of the statistics. So anything we show here will be this first forward direction above and beyond the reverse direction. And we find that there are regions that are changing on this timescale, and it's in particular this parahippocampal cortex region. So here's the whole brain version where you can see that we get this hotspot in parahippocampal cortex, and our ROI version shows this as well. That there's in this very anterior region, in the ventral visual stream, just before you get to the hippocampus, you're getting these rapid shifts as a function of the recent statistics that you've seen. This effect was marginal in CA1, which is interesting to us, but it doesn't seem like the hippocampus is really driving the effect here. Okay, and then we found that the more the recent structure mismatched the longer term structure of the data set co-occurrences, the more representational change we saw on this timescale, which suggests that the brain is doing more updating when there's more to learn, more to update. Okay, so to summarize this section, we found that parahippocampal cortex contains these semantic object representations that rapidly shift according to the recent statistics of the environment. We think this builds nicely on findings that the parahippocampal cortex represents long-term associations among visual objects by showing that there are these rapid online tweaks that are happening in these representations. CA1 was unique in the hippocampus for containing semantic representations and showed some hints of this rapid learning as well, which we think is consistent with the general way that we're thinking about that region. And then together with the long timescale drift findings, the results suggest a kind of a gradient of plasticity across these regions where, like, as you get to faster learning, you're kind of moving more anteriorly. And so this finding and others have really got us thinking about this idea that it's not just hippocampus and cortex, but that maybe there's a broader continuum here where as you move more anteriorly in cortex, you're also seeing kind of a change in these properties. So I just showed you examples of plasticity, but there's also some work on changes in sparsity as you go up the hierarchy. So if this is true, why might you organize the system in this way? And one way of asking that question, like why should it be organized this way, is to ask, well, what would happen if we took a deep neural network and allowed it to optimize for these properties? So what would happen if we allowed a model to learn its own learning rates and sparsity? Would it come up with this kind of organization? So this is a line of simulation work where we are going to allow models to meta-learn layer-wise sparsities and learning rates. So we took a variety of model architectures and a variety of data sets. And we find that the principles seem quite broad, actually surprisingly to me, across these different data sets and architectures. So like deep feed-forward, fully connected networks of different depths, and then convolutional networks. And the way that we train the way that we kind of allow the model to learn these hyperparameters is to say that each of these projections between two layers will have one learning rate that will be common to all the connections in that layer. And that rate can go up or down, right? So the model is going to be allowed to choose to make this, the connections coming into this bottom layer more plastic and the connections above less plastic, or vice versa. And then we're also going to allow it to learn layer-wise sparsity. So each layer has, some number of units that are naturally active. We're going to use a kind of a winner-take-all approach to allow the set point on the number of units that could be active to vary. So the model could choose to have few units active or many units active in that particular layer. So we adopted this meta-learning approach that was described in this Gupta paper to learn different kinds of parameters, but it worked well for us to learn the learning rate and sparsities here. So the way it works is that we alternate between inner updates and meta updates. During the inner updates, the weights of the model are adjusted as usual to optimize performance on data from a current batch of that task. And then a meta loss is computed based on the performance of the inner updated model on a mixture of current and past data. And then gradients from this meta loss are used to update the pre-inner update weights, as well as those layer-specific learning rates and sparsity parameters. So the model's asking, how can I do better with different hyperparameters, and optimizing for those at the same time that it's optimizing the weights. So we find that meta-learning sparsity and learning rate results in better performance overall, so that's good. And then I think more interestingly for us, it results in more efficient representations overall. So it's just using, like, averaged across all of the layers, it's just using fewer units to solve these tasks. Okay, but here's our key question. So what happens in the choices for these parameters across layers of the network? What we find is that as you go higher up in the network, the model chooses to have fewer and fewer units active. So things are getting more sparse as you get higher up. And then for the learning rate, we find that it's choosing to make things faster and faster, so more and more plastic as you go up the hierarchy. And there's this cool kind of critical period effect that we see here. So this is across the course of this optimization. Early on, you have the highest plasticity in the earliest layer, but that then stabilizes. So you figure out what you want to do with that first layer, and then you keep it stable as you continue to learn, and then you kind of increase the plasticity in the higher layers as you go. This also, the same kind of pattern played out in convolutional networks, and also in fully connected networks of different depths, which I don't have here. And then the last thing I wanted to show you with this work is that we were, so those are examples of kind of how do you choose these parameters across a serial organization of these layers. But we're also very interested in what happens across parallel pathways, because that's a kind of analog for what we're interested in the hippocampus. And so here we took the kind of a simple, like, fully connected input set of layers like we had before. But then at the end, we're going to divide it into two parallel pathways. the only difference between the pathways is that one will be larger than the other. And the question is, what will it learn to assign in terms of the sparsity and learning rate, the two different parallel pathways? And we find that it decides to make the larger layer sparser and faster, which is exciting to us because that corresponds to the way that we think about the trisynaptic pathway. So we get the emergence of those kind of different properties in the way that we would expect from our thinking about the hippocampus. So meta-learning, layer-wise sparsity, and learning rates results in this brain-like macro-organization, which suggests that this organization might be optimal in some way for behavior. We modeled this here as a concurrent learning process, right? So you're learning the task at the same time that you're learning the hyperparameters. We are kind of agnostic about how we think that the meta-learning process might actually be implemented. So it could be an evolutionary timescale learning process. Perhaps it's a developmental timescale learning process, like we saw with that critical period finding. So if people have thoughts on that, we're kind of actively thinking about that. But it suggests that there's some kind of meta-learning optimization that could give rise to this kind of organization. Okay, can I just do a time check? I have like maybe 5-ish more minutes of material. Should I keep going? Okay, cool. 4 minutes? Okay. Okay. So all of this has been about kind of this feed forward pass, right? So how do you encode new information as you're moving up the system? And then half of my lab is kind of interested in the opposite direction. So for information that is, initially encoded successfully in the hippocampus, then what? How is it ultimately integrated into these existing neocortical networks? So it's kind of the opposite direction. So I'll just tell you about one project in this space that we're excited about that tries to address this question of how could sleep and replay serve to integrate hippocampal and neocortical learning. So how do we integrate new and old knowledge? So we had a neural network model of autonomous hippocampal cortical learning during sleep that said that interleaved reactivation of new hippocampally dependent and old cortically consolidated information supports graceful knowledge integration over time. There are some other papers that I think make a similar kind of argument where the theoretical work that shows that new information that's inconsistent with prior knowledge requires interleaved training of old and new information. So we thought we would try to test this idea in a sleep study in the lab. So here's the satellite stimuli again. So here we're going to teach people about these categories of satellites. In this case, we've arranged them into these hierarchical structures. And we have a knowledge building phase where participants learn about the satellites over several sessions. So they are really getting a good understanding of those objects. And then we bring them back a couple weeks later, and we show them new members from a different branch of the hierarchy, but from those categories that they had learned earlier. And then they take a nap in the lab. And we use a technique in the lab called targeted memory reactivation, where we try to have some control over what people are reactivating during sleep. So in this case, we have people hearing the spoken names of the satellites while they're learning about them when they're awake. And then we quietly play those names while they're sleeping. At the kind of precise moments that we know that endogenous reactivation occurs, to try to encourage them to think about this object instead of what they might have endogenously chosen. And this technique has been used now for decades in this literature and is really quite effective at encouraging replay and changing those specific memories. Right. So we're monitoring EEG activity in real time, waiting for the peaks of slow oscillations and playing our sounds exactly at those moments. So we have 3 TMR conditions within each participant. We have one category where we're going to interleave cueing of old and new objects, another category where we only cue the new objects, and then a third category that doesn't get any cueing at all during sleep. And at the end, we do this sorting assessment where we let people group the names of the satellites in whatever way they feel makes sense to them. And the question is, do they group the satellites by the context they learned them in? So like today versus a couple weeks ago, or do they group them by the category, which would suggest that they've integrated the new exemplars into their category knowledge? And what we find is that they do both. I mean, we see effects of both the context and the category, but the interleaved cueing in particular leads people to group the old and new exemplars based on their category more so than based on when they were learned. So the category integration becomes stronger than the context separation specifically in the cued interleaved condition. Okay, so that's some evidence that interleaved reactivation of old and new information during sleep can promote that integration of new and existing knowledge. Just in case people are interested and want to check out other things we're thinking about in the sleep domain that I didn't have time to talk about today, we have a paper looking at how replay results in transformation of object memories as opposed to simple strengthening. We have some new evidence that replay results in an abstracted form of a memory using a transfer learning paradigm. And we have a memory-focused model of the detailed characteristics of rodent replay, so forward and backward replay of place cell sequences and awake and sleep replay. And so if you're interested in that, you can check out that paper. Okay, but I will give you my overall summary, and then I'm excited to take your questions. So the hippocampus, we think, might contain these distributed representations that complement the classic pattern separated representations, and this should be useful for many forms of rapid novel learning requiring integration across experiences. But when learning involves tweaks to your existing knowledge, we think that fast learning neocortical areas likely play a more direct role, and that ultimately we maybe should be thinking of this as a kind of optimized continuum of learning speed and sparsity running from early sensory areas up into the hippocampus. And then finally, that interleaved reactivation of new and old information during sleep might be useful for this process of integrating new information with our prior knowledge. Okay, thank you.